Hello everyone! Today we will be discussing the research surrounding menopause-induced obesity. Obesity develops when energy consumption exceeds energy expenditure and is defined as the accumulation of excess body fat to the extent where it causes health complications and reduces life expectancy. Obesity is commonly defined using body mass index, usually referred to as BMI, which is a measurement of body fat based on height and weight. A normal BMI is between 18.5 and 25, while between 25 and 30 is defined as overweight. Clinical obesity occurs when your BMI surpasses 30. 29% of Australian females over the age of 18 are overweight, with higher rates in women between 55 and 84, and hence understanding the impact of menopause on obesity is extremely important. So, what is menopause? The menopause transition begins with the onset of menstrual irregularities and ends with the last menstrual period. Women usually experience this between 48 and 55 years of age. It is accepted that changes in the hormonal environment at menopause are associated with an increase in total body fat and an increase in abdominal fat. But is menopause itself a cause of obesity? There is lots of controversial research surrounding this topic. Many cross-sectional studies have shown an increase in BMI across peri- and postmenopausal women compared to premenopausal women. However, the controversy arises with studies that have shown similar increases in adiposity in men of the same age. This means the changes may be due to chronological aging and reduced energy expenditure, rather than hormonal changes and reproductive aging. A prospective study by Guthrie and colleagues measured suprailiac skin folds, waist circumference, and hip-to-waist ratio in different groups of women across a 5-year period. The study found all parameters increased during the menopause transition. However, all groups gained weight, including those who remained premenopausal. There were no significant differences between the weight gain of women, supporting the idea of chronological aging rather than reproductive aging influencing body weight. The most significant gains in skin fold measurements were the cohort that became postmenopausal over the 5-year study, representing a gain in abdominal fat and differences in fat distribution. This is supported by other studies which have shown postmenopausal women have greater amounts of intra-abdominal fat compared to premenopausal women. Additionally, perimenopause leads to a rapid increase in fat and redistribution of fat to the abdomen. This is thought to be due to estrogen deficiency. So what does estrogen have to do with obesity? It is known that sex hormones strongly influence body fat distribution and adipocyte differentiation. Men have less total body fat on average, which is located around the abdomen, while women have more total body fat that accumulates in the gluteal, femoral, and subcutaneous areas. As discussed, the decrease in estrogen levels associated with menopause increases abdominal fat. However, it is not clear whether absence of estrogen is associated with the onset of obesity in women. The role of reduced estrogen levels in menopause causing abdominal fat gain has been studied using gene knockout mice and CT scanning techniques. Studies with aromatase gene knockout mice, which have no endogenous estrogen production, and estrogen receptor knockout mice, shows the role of estrogen in regulating the uptake of lipids from the circulation. Reduced estrogen receptor levels in adipose are seen in obese individuals. Further, estrogen upregulates the receptor in subcutaneous tissue, leading to higher fat storage in this area, as observed across women. Estrogen plays key roles in increasing lipoprotein lipase activity and lipolysis in the abdominal region. This effect is observed through studies which show hormone replacement therapy did not prevent weight gain, but did minimize the shift from gynoid to android fat distribution. Conclusions from this study stated that estrogen mediates this transition that is observed with menopause. Another potential mechanism by which menopause may cause obesity is the role of estrogen in appetite regulation. Estrogens act on POMC neurons, which display the estrogen receptor and cause suppression of food intake. Deletion of the receptor in POMC neurons in mice leads to excessive food intake. Estrogen has also been shown to possibly decrease ghrelin activity and reduce melanin concentrating hormones, leading to decreased hunger and less promotion of food intake. Whilst ovarectomy in mice has been shown to increase food intake, this does not fully account for the development of obesity. Decreases in estrogen levels leads to a reduction in energy expenditure and subsequent increase in abdominal body fat. Estrogens have a role in regulating many types of enzymes involved in glucose and energy metabolism, as well as lipoprotein lipase. Lipoprotein lipase breaks down plasma triglycerides into free fatty acids and glycerol. The promoter region of the gene contains estrogen response elements, and therefore estrogen regulates the activity of this enzyme via influence on the transcription of the gene. Mouse models have greatly increased our understanding of the role of estrogen in obesity. A study using ovarectomized mice showed reduced energy expenditure similar to that in estrogen-deficient women. Deletion of the estrogen receptor in mouse models has shown that mice become obese and have decreased energy expenditure, supporting the idea that estrogen receptor regulates mouse energy metabolism. Loss of the estrogen receptor in the CNS has been shown to induce excessive eating and decreased energy expenditure, while stimulation of the receptor leads to reversal of weight gain and normalization of appetite and energy expenditure. As already mentioned, estrogen regulates fat mass through promotion of both visceral lipolysis and subcutaneous adipogenesis. A study of postmenopausal women showed a correlation between fat mass and blood estrogen levels, where there was an increased fat mass with declining estrogen. However, it should also be mentioned that abnormally high estrogen levels are also associated with obesity. The most likely mechanism of this change is a downregulation of the estrogen receptor in adipocytes in high fat mass individuals, and hence a reduced estrogen sensitivity. While results from animal studies may not translate to humans due to species differences, similar responses are seen when comparing menopausal women and ovarectomized mice. These models show us how menopause may affect weight gain in women. In conclusion, analysis of the current research has shown that weight gain itself is unlikely to be affected by the hormonal changes of menopause, but rather lead to a change in body composition and the accumulation of central abdominal fat.